Hi, I am Aurel Enriquez, and this presentation contains our discussion on week 12, which focuses on the laboratory diagnosis of viral infections. Now, the topic of laboratory diagnosis of viral infections and actually clinical virology in general, this specific field of medical science continues to be super interesting but very challenging at the same time because new viruses or new strains of previously existing viruses are continuously being discovered, and even the diagnostic methods, there are new diagnostic methods that are being developed. So there's a constant change, when we look into the perspective of studying clinical virology. Now, in most older or traditional diagnostic methods, a lot of them were seen to be quite time-consuming, all right? And some of them, kailangan pa ng specific trainings or kailangan pa ng ibang personnel to perform these different traditional diagnostic methods, which would include microscopy, specifically yung electron microscopy and cell culture. These ones are super time-consuming and not that easy to perform. Serology has always been there, no? Even until now, na may bagong methods na na-discover. Serology is still one of the widely used methods in diagnosis of viral infections. Pero yun nga po, no, kasama na siya sa listahan ng mga traditional diagnostic methods. Now, with that, no, the main methods of diagnosis that has been, or that is actually constantly being developed until today is the molecular diagnostic methods. Now, the advantage, to these molecular diagnostic methods is that they're much faster, they're more sensitive, and more specific, which would lead to earlier interventions. No? Mas maaga pong madadiagnose ng tama yung pasyente, so mas maaga rin mabibigyan ng tamang course of treatment yung patient. So that would lead to more favorable patient outcomes. So kumbaga, the prognosis would be better for the patient because the diagnosis is quicker, so the solution or treatment would also be given at an earlier time in comparison to other methods such as cell culture. So although cell culture has been kind of like the standard method, it's still very time consuming, which is why ayun nga po, mas favored na yung mga recently developed molecular diagnostic techniques. Let's proceed with our discussion on specimen selection, collection, and transport. So, paano natin malalaman kung ano yung tamang sample to collect? How do we collect these samples? Anong materials yung kailangan natin? And what are the proper transport guidelines? For full-service virology laboratories, so yung mga tertiary level type of laboratories, or maybe those laboratories that are referred to as reference labs, and even those types that are considered as research labs. Now, these are the types of virology laboratories that offer detection methods such as cell cultures, all right? But again, this is only available for full service virology labs. Now, for routine laboratories, or the ones that we're more familiar with and we're more, or it is more accessible for most of us, in routine laboratories, what they usually offer is viral serology. So usually these are the types of tests where you could collect blood or other samples from the patient, tapos ito-drop lang nila dun sa test kit. So you just have to wait for a certain number of minutes, or you would just add a certain buffer or other stuff like that, and then after a certain number of time, maybe like 10 minutes, 20 minutes, results will be available. Yun po yung usually available, sa karamihan ng mga routine laboratories. Viral serology through testing kits. All right. So the clinical symptoms usually point to the target organs involved, and this would determine the appropriate specimen. So ngayon, isipin natin, paano natin malalaman kung ano yung tamang sample to collect, depending on what infection the patient is currently suffering from. Now, when deciding which specimen we should collect for a suspected viral infection, usually Ulitin natin, the clinical symptoms would point to the target organs that are involved or that are currently being targeted by the virus. So, if we pay attention to the clinical manifestations, this would guide us on which sample to collect. And subsequently, this would lead us to choosing the appropriate diagnostic methods and giving us an idea on which possible infecting organism we should expect. So, di ba po, by now, common sense na sa atin to, no? Na nakatatlong fields na po tayo ng microbiology. Simula nung time nyo sa parasitology until you went through bacteriology. We just finished mycology last midterms. So, hanggang ngayon po sa virology, no? Common sense na ito para sa atin. So, kung pulmonary symptoms po yung clinical manifestations ng pasyente, what do you think would be the appropriate sample? Respiratory samples, di ba po, such as sputum, nasopharyngeal swabs, or maybe bronchoalveolar lavage fluids. Kapag CNS symptoms, no, symptoms involving the CNS naman po, ino-observe natin sa pasyente such as recurring headaches, blurry vision, loss of control over motor functions. Ano po kaya yung appropriate specimen for those types of cases? Most likely cerebrospinal fluid, di ba po? And other supporting tests would be performed, such as CT scans. So, ganun lang naman po, no? That's how specimen is collected based on the clinical manifestations to help us correlate everything with the possible causative agents. So, again, usually, in routine laboratories, viral serology po yung pinaka-common na diagnostic method na available. So, viral serologic tests, lalo na po yung mga rapid diagnostic tests, they are inexpensive. They're not very pricey, and they're actually easy to perform. Right? So examples for these would be the hepatitis B surface antigen testing or yung HBsAg to check for viral hepatitis. That's one example of viral serology. Another example would be the dengue IgM and IgG testing kits, diba? So for those types of tests, serum lang po from the patient yung kailangan, i-drop lang doon sa sample well yung nung test kit and then read after an indicated amount of time. Pero yun nga po, no, ulitin natin, for full-service virology labs or mga reference labs and research centers available sa kanila, no, yung iba pang mga viral diagnostic techniques na mapag-uusapan natin mamaya, and lalong-lalo na yung cell culture. So we'll just discuss that later. Next, let's talk about viral shedding and knowing when to collect a sample. All right, now because viral shedding is greatest during the early stages of infection, the best specimens are those that are collected as early as possible. All right, so maximum or matagal na po yung 3 days or 5 days after the onset of symptoms. All right, so dapat po as soon as the symptoms are being observed, no, already start collecting samples. Now, how would collecting the sample at a much later time negatively affect the diagnostic test results? So, kung madadali tayo sa pag-collect ng samples, ano naman, di ba? Ano namang negative effect nito? Now, according to our reference textbook, no, the sensitivity of viral culture can decrease rapidly in just 3 days after the acute onset of symptoms. So, possible po na kahit present naman talaga yung suspected virus and yun naman talaga yung causative agent ng disease or clinical manifestations ng patient, magne-negative na po sa culture yung sample ng patient because their numbers or the patient's viral load have significantly decreased in that span of time that the sample was not collected. No? Kasi po, nabigyan na ng time yung immune system natin na mag-react and to fight off the pathogenic virus. So, bumaba na yung amounts nila. Kaya po, mahalaga, no, that during the early stages of infection or less than 3 days after the onset of symptoms, dapat nag-collect na po ng sample. Now, let's talk about swab specimens. For swab specimens, the appropriate swab material that we should use would either be Dacron or Rayon. So, we should never use swabs that are made up of calcium alginate. No, bakit po? Because Calcium alginate swabs can inhibit the replication of some viruses, and they could also interfere with nucleic acid amplification tests. All right, so this photo right here shows us how a full set of commercially available viral diagnostic swabs would look like. So usually yung manufacturer na rin mismo, no, yung nagsaset na Dacron or rayon lang talaga yung gagamitin nila na material. Yung mga manufacturers na rin mismo yung sinasadya nila na iwasan na yung calcium alginate, lalo na kung minamarket nila yung transport media or collection material, no, for viral test or viral diagnosis. Now, the different types of specimens may be transported using different transport media, but in general, no, you should be using sterile containers to avoid contamination. Let's talk about that on the next slide. Different types of specimens may be transported using different transport media, but in general, it should be sterile to avoid contamination. And it is preferred that they are screw cap or yung iniikot po yung takip so that we could close them tightly and avoid leakage or drying of the specimen. Now, speaking of contamination, minsan po may mga components din itong mga viral transport media na to that are antibacterial and antifungal just to eliminate any possible contaminating organisms from the environment or from the patient's normal flora. Now, it is best to process viral culture specimens immediately, but if they can't be processed immediately after collection, then they should be stored at 4 degrees Celsius if it's only going to take a few hours. Pero kung araw po, no, yung bibilangin, kung ilang araw yung bibilangin, we would have to wait for days before we'd be able to process the sample, then the required temperature should be at negative 70 degrees Celsius. Again, negative 70 degrees Celsius. We should never store these samples at negative 20 degrees Celsius because at this temperature, this would facilitate the formation of ice crystals. Now, these ice crystals will disrupt the host cells and result in the loss of viral viability. Okay, so tandaan po natin. Okay, tandaan natin, virus particles can only survive for as long as they are inside of the intact host cells. So, if masisira yung host cells, no, the host cells would be destroyed due to ice crystal formation, hindi rin po magtatagal, the viral particles present in the sample would also cease to be viable. All right, so again, for if it's only going to take a few hours, the preferred storage for viral transport or viral samples should be at 4 degrees Celsius. But if it's going to take days or weeks or for long-term storage, you should keep the samples at negative 70. Keep in mind to never store your viral samples at negative 20 because of ice crystal formation. Let's now proceed to enumerate what the appropriate specimens would be for maximum recovery of the virus of interest, depending on which organ system is being affected. So as we can see right here on table 29-2, makikita natin na naka-arrange na po sila depending on the organ system or body system that is affected and kung alin po yung mga specific viruses na pwede nating ma-detect depending on what diagnostic test we would use. So let's break this down in a format that's a bit more easier to understand. On this slide, ito po yung magiging guide natin. Alright, so each of the following slides are focused on one specific body system. And then under that, we'll see the diseases associated or maybe the clinical manifestations that we may be able to observe. After that, we would look into the acceptable specimens in this scenario. All right, so again, based on the diseases associated, what would be the acceptable specimens? And lastly, we would look into the possible causative agents that we can detect from these clinical samples. In cases of pneumonia, bronchiolitis, or croup, ito po yung mga specimens na dapat nating i-collect. Just a side note, no, just in case you already forgot, croup refers to an upper respiratory tract infection which would obstruct breathing and could cause a characteristic barking cough. So, di ba minsan may na-observe tayo mga inuubo, tapos parang tumatahol yung tunog ng ubo nila. So, that's what we mean when we say croup. Now, acceptable specimens for cases like these would be nasopharyngeal swabs or bronchoalveolar lavage fluids. And ano naman po yung mga viral pathogens that we can expect to detect from these samples? The most common ones would be influenza and parainfluenza viruses, the RSV or the respiratory syncytial virus, and simula po nung late 2019 up until today, kasama na po sa listahan ang mga coronaviruses. Now, what are the specific examples of coronaviruses? Magdi-discuss naman po natin to ulit pagdating natin sa week 15. But anyway, no, maliban po sa SARS-CoV-2, now, of course, meron din yung SARS-CoV or the causative agent of severe acute respiratory syndrome, which became an outbreak no, back in 2002. So, bale, parang siya yung OG ng mga SARS-CoV. Another example would be the MERS-CoV or the Middle East Respiratory Syndrome coronavirus, which is primarily a zoonotic virus, but this one also became kind of like a public health concern somewhere around the earlier half of 2019. So yun po yung mga examples no, ng coronaviruses na pwede nating ma-detect using nasopharyngeal swabs. And of course, yung pinakabago po, yung kilala nating lahat is SARS-CoV-2, the causative agent of COVID-19. For patients suspected to be suffering from viral gastroenteritis, there are diagnostic tests that make use of stool samples or rectal swabs or buccal cavity swabs. Now, the expected causative agents would be rotavirus, Norwalk virus, or the adenoviruses 40 and 41. So next week po, pinakauna nating madidiscuss, no? So week 13, pinakauna nating madidiscuss ang adenoviruses. Now, people may be wondering, no, kung gastrointestinal yung symptoms, bakit kasama sa listahan ng possible specimens ang buccal swab? Because we're considering the point of entry of the causative agent, diba? A lot of these viruses that are capable of causing gastrointestinal symptoms via ingestion or through the fecal-oral route po yung mode of transmission nila. So buccal swabs could be helpful, no, considering the mode of transmission or point of entry of the organism. Kaya po kahit gastrointestinal yung symptoms, kasama sa possible sample yung buccal swab. Cutaneous viral infections would require the collection of vesicle aspirates, lesion swabs, and nasopharyngeal aspirates. Nasopharyngeal aspirates could also be helpful kasi po for some viruses, although their clinical manifestations could show on the skin, no, yung mode of transmission po nila would begin via inhalation of the virus particles. So it would make sense for us to also collect samples where we think the virus entered the host. Now, the possible infecting agents for cases such as these would include human herpes viruses 1 hanggang 7, no? And except the Epstein-Barr virus, which is HHV4. Other causative agents would include the parvovirus and the Coxsackie virus. For these next ones, let's first differentiate what is conjunctivitis and what is keratitis. Now, conjunctivitis, this is the infection of the conjunctiva, which is a transparent membrane that lines your eyelids and covers your eyeballs, all right? Now, this transparent membrane still does have capillaries across them, so when there is any infection or inflammation, ito po yung nakikita natin. So, merong redness, no, namumula yung mata ng pasyente, as we can see right here on the top left picture. Again, this is an example of conjunctivitis. Now, when we say keratitis, this is an infection or inflammation of the cornea, which is also clear. Pero ito po, it only covers the iris and pupil specifically. Alright? So ulitin natin, no? Sa conjunctivitis, yung buong lining that covers the inside of our eyelids until the entire eyeballs, no? That's what's being affected. But for keratitis, yung specific area lang that covers the iris and the pupil, that's the only infected part. Now, again, let's look at this photo right here. No, this photo right here shows us the an example of epidemic keratoconjunctivitis, which is a combination of both. All right. So kapag sinabi natin keratoconjunctivitis, both the conjunctiva and the cornea of the patient is being affected. Now, acceptable specimens for viral keratoconjunctivitis, which or this would include conjunctival scrapings and corneal swabs. All right, and then possible causative agents would be human herpes viruses 1 to 4, and then there are also some strains of adenovirus that could cause keratoconjunctivitis. Next are infections involving the central nervous system. So that would include aseptic meningitis or viral encephalitis. The acceptable specimens, of course, would be cerebrospinal fluid. But again, let's look at this list right here. Bakit kasama ulit yung nasopharyngeal swab? Hindi naman respiratory yung symptom. Because some viruses can cause CNS infections, may have possibly been acquired by the patient when it entered the respiratory tract. All right, so again, for CNS infections, we're also considering the mode of transmission. So again, for CNS manifestations such as aseptic meningitis or encephalitis, acceptable samples would include cerebrospinal fluid and nasopharyngeal swabs. Dahil nga po, maaaring yun yung point of entry ng organism. Okay? So, ayun po. Suspected viral agents would include human herpes viruses 1 to 3 and arboviruses. Now, when we say arboviruses, the word arbo is actually a contraction of two different words. So it actually means arthropod-borne. Okay, arboviruses, arthropod-borne viruses. Okay, so arboviruses or arthropod-borne viruses, this means that their mode of transmission is through the bite of an infected arthropod vector. So it could be a flea, a tick, a mosquito, or any other insect. All right, so number one example for this would be the dengue virus. So in some cases, dengue infections could lead to encephalitis. Genital infections could manifest as either urethritis or genital lesions, and the appropriate specimen for these would be aspirates or swabs from the vesicles that have formed on the patient's genital area. The primary suspects would include human herpes viruses 1 and 2. Disseminated viral infections could lead to viremia, or the presence of the virus on the patient's circulating blood. So the acceptable specimens would be whole blood collected within EDTA tubes, or pwede rin namang serum or urine samples. Now again, people might be asking, bakit kasama sa listahan ang urine sample kung viremic ang pasyente? Remember, the main function of the kidney is to filter our blood. So chances are, if the virus is present on the patient's blood circulation, or kung halimbawa yung mga antigens niya, the antigens or maybe the antibodies that were produced against the infection, hindi po malayong mangyari na ma-filter out din ito nung kidneys nung pasyente, which are now going to be shed in the patient's urine. Alright, so ayan po, we're considering that either the antigens of the virus or the antibodies that were produced against the virus would be filtered out by the kidneys and would show up in the urine samples. Kaya po, kasama sa choices of acceptable specimens ang urine samples for disseminated infections. Listed here are the viral pathogens capable of causing viremia, human herpes viruses 1 to 4, hepatitis B virus, and arboviruses, or yung mga arthropod-borne viruses. Last on our list are viral organisms that could cause congenital infections. So, ang mga samples po na pwede nating i-collect in these cases would include throat swabs, serum, or urine. And the viruses that are capable of causing congenital infections would include CMV, or the cytomegalovirus, HSV, or the herpes simplex viruses, and rubella. Let's proceed to the next segment of our discussion, wherein we would go through the different viral diagnostic methods. Now, para po sa inyo, no, for undergraduate students, very concise lang din po yung discussion on viral diagnostic techniques, because a lot of these would be kind of like an overlapping discussion for your immunology classes. Another thing is that hindi rin naman po kasi commonly performed yung karamihan sa mga ito. Alright, so not a lot of clinics offer these diagnostic methods, and most of these sa mga research institutions lang pine-perform. So let's just quickly go through some of them. These first few methods involve the direct detection or visualization of the virus from clinical samples. Keep in mind, though, that direct detection methods are only presumptive. Okay, and also they're not as sensitive as culture methods, but they do offer quick results para na rin po mabilis nang makapag-decide yung physician on which treatment protocol would be best for the patient. So first up is microscopy. All right, now the most commonly used method would be the bright field microscopy. So ito po yung ginagamit natin after staining the sample and observing for the cytopathic effects. We'll talk more about the cytopathic effects later. Okay. Now, electron microscopy is not a very widely used method dahil electron microscopes are expensive, and electron microscopes could only be handled by people specifically trained in electron microscopy. So, hindi po lahat ng medtechs, no, lahat ng medical technologies, hindi po tayo usually yung nag-ooperate ng isang electron microscope unless we have a training for that. And although visually na nating makikita yung virus, its specific species or strain would still have to be confirmed in conjunction with other methods, specifically molecular diagnostics. Kaya po usually dumadiretso na lang sa molecular diagnostic methods yung mga laboratories. Alright? So primarily ginagamit lang po ito ng mga research institutions in helping us visualize and identify novel or emerging pathogens. Next on the list is immunofluorescence. So we'll talk about that in the next few slides. But let's first get into the cytopathic effects. All right, so CPEs or cytopathic effects, these are visual changes to the affected cells. All right, and different viruses could produce different cytopathic effects. So let's see how this would look like on the next slide. Here we can see the different types of CPEs na pwede nating ma-observe using a bright field microscope and the appropriately stained tissue samples. Alright, so there are three types. First up are intranuclear inclusions. As the name implies, they can be observed within the nucleus of the cell. So right here we have the Cowdray type A bodies that are produced by the varicella zoster virus, and this is what they look like. Next up are the owl eyes. All right, so owl eyes inclusion bodies, they are produced in cases of cytomegalovirus infection. So they look like this one, as we can see here in this second photo. Ang next classification po ay cytoplasmic inclusion. So cytoplasmic inclusions, nasa labas na siya ng nucleus. All right, so makikita na natin to anywhere on the cytoplasm. An example of that would be the rabies virus. So the rabies virus produces what are called negri bodies, all right, which was first discovered by the Italian pathologist Aldecchi Negri. All right, so hindi po Hindi po descriptive ng ibang bagay ang Negri bodies. This is not a word. This is the last name of the person who discovered these structures. So again, the Italian pathologist Aldechi Negri discovered these eosinophilic structures found within the cytoplasm of the nerve cells of rabies-infected animals or humans. All right. Up next are nuclear enlargement. All right, so nuclear enlargement, this is otherwise referred to as coilocytosis. Okay, so coilocytosis, this is typically caused by the human papillomavirus, as we can see right here on the fourth photo. May isa pa po na hindi kasama dito sa listahan na to. It is referred to as syncytia formation. Now, syncytia formation is caused by the respiratory syncytial virus or RSV. This will further be discussed in the next few weeks or in the following weeks. All right, so hindi lang po nakalista dito, but another would be syncytial formation, which is caused by the respiratory syncytial virus. Still on microscopy, we got immunofluorescence techniques, specifically direct fluorescence antibody. Now, immunofluorescence can be a valuable tool to detect various viral agents directly in clinical specimens. We also have the direct fluorescent antibody. So direct fluorescent antibody, the cells from a patient are fixed onto a microscope slide, and then fluorescence labeled antibodies are added to that. All right, they're added to the sample. Now, if the viral antigens are in fact present on the patient sample, these labeled antibodies will bind and cause fluorescence, and we would be able to see that microscopically. All right, so the photo on the top left is an illustration on how it works, and then the example on the bottom right, yan po mismo yung makikita natin if we were able to perform the procedure correctly. Again, this is referred to as direct fluorescence antibody. Next are enzyme immunoassays. So many enzyme immunoassay tests for viral detection are commercially available, and most of them would make use of multi-well microtiter plate assays, as we can see right here in these photos. Now, these tests can detect RSV and influenza A from respiratory specimens. Pwede rin makapag-detect ng hepatitis B virus and HIV-1 from serum or plasma. You could also detect enteric adenoviruses from stool samples and herpes simplex viruses from cutaneous lesions and conjunctival swabs. Alright, so dito po natin pwedeng gamitin yung karamihan sa mga appropriate specimens that we have enumerated on the earlier part of our discussion. So please do not forget to study the table that was included here. Nucleic acid-based analysis have the capability to determine the presence or absence of a virus, and also, depending on which assay is used, it could also give us a quantitative result. All right. So, for example, an example of that would be the real-time polymerase chain reaction, or the RT-PCR. Now, of course, there are advantages and disadvantages to nucleic acid-based detection. So advantages would include a much faster turnaround time. So mas mabilis yung results, no, gamit itong nucleic acid-based methods na to. Next, we also have better sensitivity in comparison to cell culture and direct fluorescence immunoassays. And these assays could also be quantitative. Alright, so detection of viruses that are non-culturable by cell culture is also an advantage. Anong ibig sabihin nito? May mga viruses out there po kasi, no, that we cannot grow them in cell culture. Okay, hindi natin sila kayang i-grow in the laboratory. So instead of doing that, we could just move to nucleic acid-based detection. So examples of non-culturable viruses would include the noroviruses or the hepatitis viruses. So, yun po yung ibig sabihin when we say non-culturable. Hindi sila kayang i-grow in cell culture. So, i-detect na lang natin yung nucleic acids nila. So, these nucleic acid-based tests are also able to detect multiple viruses at the same time. Alright? So, lalong-lalo na po kung multiplex yung type of test na gagamitin nyo. Alright? So, and potentially, they could also characterize the virus genetically kung i-undergo nyo ito ng genotyping or kung i-undergo nyo ng genotyping yung product of amplification. But of course, no, that goes with a few disadvantages. So, higher cost, medyo mas expensive po yung mga ganitong methods, lalo na yung mga consumables. There is a need for specialized training. So, again, hindi lahat ng medical technologies agad-agad capable for nucleic acid-based testing. No? You need a specific training, a specific certification to perform these types of tests. And of course, these types of tests must first be cleared by the FDA, or here in the Philippines, no, dapat may go signal muna ng Department of Health. So listed here are the different examples of nucleic acid-based detection. PCR with flow cytometry, pwede rin yung hybridization assay, RT-PCR, and the digital PCR. Next is viral isolation. In clinical virology, isolating viruses is still the gold standard against which all other methods are compared. So meron pong 3 different methods on how we could isolate viruses. So first up is animal inoculation. So animal inoculation is extremely costly, all right? And this one is only used in research or resource laboratories, and it's highly subjected to ethical concerns. So this photo right here shows us intracerebral inoculation that is being done on a laboratory mouse. It's actually quite sad if you keep looking at it long enough. Like, I know these mice were bred for this specific purpose, but you know, if you're a person who loves animals, you can't help but get this weird, sad feeling kapag nakakita ng mga ganitong pictures. Anyway, next po no, is embryonated egg inoculation. Now, this one is also very rarely used, so we will no longer be discussing that in detail. But here is an illustration showing us the different sites on where we can inoculate the embryonated egg. And depending on the virus, nag-iiba rin po yung sites kung saan yung preferred na area kung saan mo dapat i-grow yung virus. Okay? So, next to that, no, on the next slide, mag-focus po tayo sa cell culture. The term cell culture is technically used to indicate a culture of cells in vitro. Okay? So the cells are not organized into a tissue. So naka-plate or naka-spread out lang sila sa isang media, no, yung mga live cells, but the media is not necessarily a tissue sample. Okay? So sana naintindihan po, no? We're not using actual tissue samples. We're only using a culture media with living cells in it. But again, these living cells are not arranged in a manner wherein they are considered as a tissue structure. Now, these cell cultures can be divided into 3 categories: primary, low passage, and continuous. So, primary cell cultures, they make use of cells that have been taken from animal tissues. Now, after the animal tissue is processed and its cells have been dispersed, these cells are then seeded onto the surface to form a monolayer or a single layer of those living cells, and they're placed inside of a flask or a test tube or tiny petri dishes, as we can see right here in these examples. All right, so ayun po, no, the downside to this one, no, ang downside lang po or ang disadvantage for using primary cell lines, no, the problem with primary cell lines is that they're only capable of undergoing minimal cell division. All right, so bakit po natin sinasabi na ito ay isang downside or ito ay isang disadvantage? Kasi po, diba, viruses, they can only survive within living cells. Now, once these primary cell cultures die or once natapos na nila lahat ng possible cycles nila of cell division, hanggang doon na lang. So, hindi na po tayo, or kung halimbawa hindi pa po tayo tapos dun sa research or dun sa observation natin, then we'd have to make a new batch of cell cultures. Okay? Next are low passage or finite cell lines. So, ang lamang lang naman nila, no? Lamang lang sila ng konti sa mga primary cell lines, pero may limit pa rin, alright? So, they can only last for up to 50 passages because they're made up of diploid cells. Third type is the continuous cell lines. So, ito po yung pinakapangmalakasan sa kanila, alright? So, these cell lines are capable of infinite passaging. So, ano po ba ang passaging and how does this help us continue using the same cell line, alright? So, passaging, this is the process wherein the cell viability is maintained by periodically removing the cells from the surface and then diluting them and then placing them into a new container. So, yung mga bagong cells na produkto ng previous cell division, andun sila sa surface, kukunin natin yon. Tapos, kumbaga is parang we'll make a new batch of cell cultures. Alright? So, ayun po, no? Hindi na natin i-discuss yung step-by-step process na yon because hindi naman yun tinatanong sa board exam. And it is also very unlikely that you'll ever do that in your entire career as a medical technologist dahil nga po, limitado lang din ang mga virology laboratories dito sa atin sa Pinas. And third reason is, ayun nga po na we're already shifting towards molecular diagnostic methods. Next, we have what is known as the centrifugation-enhanced shell vial culture. Now, shell vial culture is a technique that can more rapidly identify viruses in comparison to the traditional cell culture method. Now, the idea is very much the same. You have a monolayer of cells, and then you would inoculate that with your sample. But for this one, no, iba yung format niya, kumbaga. Now, the cells are grown on a round cover slip that is placed inside of the shell vial. Now, this shell vial is a small, round, and flat-bottomed tube, which is generally screw-cap. So, yung takip niya po ay iniikot din. So, the shell vial, which would contain the cell monolayer on that cover slip at the bottom, would be inoculated with the clinical sample. And then you would centrifuge that, no, to promote viral absorption. Now, if you would remember our previous discussion for week 11 when we talked about the viral replication method, di ba po, yung first step is adhesion, alright? Which the virus would first have to find its target cell attached to that before the next few steps would happen. Now, for the shell vial culture, what we're doing right here is pinapabilis na natin yung process ng paglalapit ng virus papunta dun sa target cell by centrifugation. So by centrifugation, you force the virus particles to move downwards papunta dun sa cell culture or dun sa monolayer ng cell. Alright, and after that, you would incubate it for 24 to 48 hours at 35 to 37 degrees Celsius. After that, you would be able to kind of like remove the cover slip at the bottom and proceed with immunofluorescence. So again, it's very much a similar concept to the traditional cell culture method, aside from the fact that, ayun nga, meron tayong vials or containers and we undergo or we subject the sample to centrifugation just to speed up the process a bit more in comparison to if we don't do that. Moving on, we've got serological assays. Now, viral serology detects circulating antibodies to viruses after the exposure, which is why it has been said to only give us a retrospective view, all right? So other limitations of serological tests include measuring only the host's response to the virus other than detecting the actual presence of the virus. So ang dinedetect lang natin dito is yung naging reaction, no? Yung naging products ng immune system ng patient against the infection. But we aren't really capable of actually detecting if the virus was in fact present. Kumbaga, yun nga, ulitin natin, yung immune response na lang yung tinitignan natin, hindi na yung actual presence of the virus. Second thing that we need to take note of is that the antibody producing capabilities of humans would vary very differently. Okay? Paiba-iba po, no? Halimbawa, yung mga taong immunocompromised or immunosuppressed. So naturally, we would expect, no, na mahina yung immune response nila. So the chances are hindi sila makakapag-produce ng detectable levels of antibodies, no? Unlike for normal immunocompetent people. Okay? So the third thing that we need to keep in mind for serologic tests is that the antibody level does not necessarily correlate with the acuteness or the activity level of the infection, because this is also host dependent. For some people, konting exposure lang, nag-react na agad yung immune system nila. For some people, no, kahit ongoing na infection, medyo mas slow yung reaction ng immune system, mas konti yung antibodies na mapoproduce, and ayun nga, maaaring mag-false negative sila dahil yung antibodies that's currently within their circulation is still not at the detectable levels of the testing method. Okay, so one of the most well-known and utilized advantages naman po ng viral serology is that some assays are capable of distinguishing between IgM and IgG. Now, the presence of IgM indicates that the infection is acute or recent, okay? And a positive IgG, no? Kung IgG naman po magpo-positive yung pasyente, this indicates that this is possibly the patient's second encounter with the virus, okay? Now, most serologic assays are cheaper and easier to perform compared dun po sa ibang mga methods na na-discuss natin kanina, which is why serological assays are still one of the most commonly used viral detection tests. Okay, now this is also useful for detecting infections from viruses that are non-culturable. So yun po, pwede din natin gamitin ang viral serology kung yung virus po na gusto nating i-detect ay hindi kayang mag-grow in culture. Okay? Another thing, pwede din natin itong gamitin to check the patient's immune status if, for example, second time na niyang magkaroon ng infection, or pwede rin naman po natin gamitan ng serology para ma-check natin if the person has already been vaccinated to the virus of interest. So vaccination, diba? This leads to our immune system producing antibodies against a certain pathogen without actually getting sick because of that pathogen. All right? So serologic methods din po yung most common method of viral detection here in the Philippines. If you would remember during yung kasagsagan pa po, no, during the height of the pandemic, rapid antigen testing for COVID-19 also kind of like became a thing, all right? And of course, di pa rin nawawala, no? Yung dengue IgM and IgG test kits, HBsAg test kits for hepatitis B virus detection, and ayun po, yun yung mga pinaka-common na examples. Other uses may include the capability to monitor immunosuppressed patients if they or if their immune system is improving or not. Pwede rin gamitan ng serology yun. And lastly, the data from these serologic assays could also be used for epidemiologic studies. Here are the references that were used in the making of this presentation. But of course, tatandaan po, ang main reference pa rin natin is Mahon 7th edition. Now, that ends our discussion for week 12. This has been Aurel Enriquez. Thank you so much for watching.